Hi, welcome to the Biodiversity Genomics Conference. This is the ASG Project talk. So this is the Aquatic Symbiosis Genomics Project. I'm the project manager, Victoria McKenna, and I'll be discussing the ASG project. So the ASG sits under the Tree of Life, which is a program to collaborate widely to deliver integrated projects across the genomic diversity. So this includes the Darwin Tree of Life project, the ASG project, the VGB project, and the Earth Biogenome project as well. So today I'll be discussing the ASG one, which is sequencing around 1,000 species from marine and freshwater. So the ASG project goals are to build a community of symbiosis researchers to support answering biological questions. And these are things like the origins of symbiosis, the ongoing evolution of symbiosis, and responses of symbiosis to environmental change. To sequence and annotate the genomes of about 500 symbiotic systems from freshwater and marine environments. And this will mean the generation of around 1,000 genomes, so the host and the symbiont together. And so this includes sequencing the holobiont itself. We offer training and support to deliver the genome bioinformatics expertise in the community as well. There's around 10 hubs, each with a biological focus in terms of taxonomy, ecosystem, or mode of association. There's about 14 partners that are working on these systems because each hub may include one or more partner. And each of these partners provides a list of species that will be collected and shipped to Sanger for sequencing. So these are the lead PIs. This is the project title, and these are the macrosymbionts and microsymbionts. The project titles, we include everything from mollusks, sponges, symbiosis, in photosymbiosis, and cephalopods as well. So the hosts or the macrosymbionts can include anything from mollusks, sponges, corals, and the microsymbionts are usually things like bacteria. So this is eukaryotic target taxa of the host. So for example, you can see here just how diverse it is, and indeed it's across the Tree of Life. This image was provided from John Archibald and Mark Blackster. So this is a really brief overview of the sample workflow that I'll be talking about today from ASG. Once the partners have collected all their samples right at the beginning of the project, they ship them to us after they've passed kind of numerous checks. So it'll be things like wildlife, environmental law, and legal checks. This will be then sent into us, and we will then carry out DNA and RNA extraction in the lab. So once the DNA and RNA extraction have passed the QC, it's then passed on to our scientific operations teams for sequencing. This sequencing is then carried out and passes the data quality assurance and is assembled as well and scaffolded. And this is assessed against our Tree of Life quality control. This is handed over to our curation team who carry out the curation manually. Annotation isn't yet happening on the ASG team, but once that happens, that will be submitted as well along with the curation. All of this is submitted to EMBL ENA database and the data portal too. So we've got our own Aquatic Symbiosis Genomics data portal provided by EMBL EBI, which helps us track the status of the species and where it is in the pipeline. All of this is published in the Wellcome Open Research portal as genome notes for every single species as well. So right at the beginning of the pipeline, we have the sample acquisition. So the partners collect the samples from all over around the world, and including their sub-hubs as well. And they're snap frozen in tubes from living tissue in nitrogen or dry house and stored at -80 or below. Before shipping, we require that all legal documentation is in place and a completed manifest which carries the metadata is fully validated. So this is things like checking the environmental, wildlife and environmental law, CITES permits, Nagoya and ABS checks are carried out and passed. This is a screenshot from our sample tracking management system, and this helps to monitor the samples that are coming in, as well as looking at things like the number of species delivered by partner and the status it is in the pipeline. For example, the legal checks are in progress here, whereas the wildlife and environmental law have been completed. So once the samples have arrived, they're passed over to our toll core lab team, and they carry out the DNA and RNA extraction. So the large samples are disrupted using cryoprep and smaller samples with a power masher, and the femtopulse is used to assess the length of the recovered DNA for quality control checks. So this is for both RNA and DNA extraction. So our sample tracking management system again helps us to look at monitor the performance of the extraction here. A good quality extraction features high molecular DNA, a high yield, with some sample tissue remaining for high seed preparation as well. So this is then handed over to our genome sequencing team, so our scientific operations team, and they carry out high seed data they're generated on Illumina platforms using the ARIMA high seed protocol. The long read data is generated using the low input library prep method and sequenced on PacBio as well. And our RNA-seq is generated on Illumina platforms. Again, we've got a sample tracking system which helps track the progress being sequenced on the different platforms. So we can see whether each species, the high seed is in progress, for example, or whether the PacBio is complete. For genome assembly, it's then handed to our genome assembly team once it's passed the sequencing QC, and the PacBio hi-fi reads are assembled using HiCanoe or HiFASM. We have different tools that assemble mitochondria and plastids. The duplications are removed, and we also have tools to help scaffold the genomes with the high C data as well. And all the data QC and the primary assembly metrics are made available on our Toll QC website. So this is an example of the giant clamp that was sent in by Joe Lopez. So our current default approach in trying to assemble the symbionts is to assemble all the reads together first and then separate the contigs by species after assembly. But we're also trying to separate out the reads of the sponges, for example, and by taxa before independent assembly. So trying to separate those symbionts out before the assembly or after the assembly is something that we're trying to work out which way isn't the best method to carry out. So the symbiont separation decontamination has been one of the most difficult steps, and we've used tools to identify and isolate the cobalt genomes from the complex metagenomes. So the assembled scaffolds are separated using sequence similarity, K-mer frequencies, and other metrics. So one of them is blob toolkit you can see on the left. So for example, the light blobs are the protobacteria, and you can see the red ones are the cyanobacteria as well. So this is then handed to our curation team who manually curate them. The assembled genomes are manually curated using the high-C maps and GEVAL. The high-C data in particular is interrogated and used to improve the assembly correctness. So on the left, you can see the high-C contact map of the giant clam that's submitted as well. These curated genomes are post-processed and submitted to ENA. So at the bottom, you can see the giant clam that's been submitted to ENA at the moment. The genome annotation we've not yet carried out on the ASG project, but it will be, and a repeat library will be generated for each species, and the protein coding genes are identified using RNA-seq to train the gene model predictors. So to make all the data accessible, we have a dedicated ASG portal, which Emily BI provided, and it accesses all the data generated by ASG, including the status tracking. So you can see for the giant clam, we've got assemblies submitted to ENA. The annotated genomes will be hosted by Ensemble, and the final annotations, including the protein domain information, will be accessible as well. So for data publication, everything that will be published will go through Wellcome Open Research Gateway. So we've got a Tree of Life gateway here, and this is our ASG. We've got our own ASG one at the moment. It's just got our published paper. The first one is in progress, which is the giant clam from Joe Lopez's group. And this one will be the first one that will be published on the gateway. A quick update. For the goal is about 500 symbiotic systems. We've got about 181 on site, about 68 in data generation or assembly, and 2 have been submitted at the moment. John Archibald's epithemia will be the first host and symbiont assembly to be completed in the project. One of our main goals is to deliver training. So we work with key partners to deliver approaches for genomics, carpentry's workshop, data analysis, train the trainer, and focus workshop, working with Connecting Science, MBI, train the trainers, carpentries, and Python biologists. So I'd just like to thank all the collaborators and contributors, so everyone from Sanger, from all the different lab teams, the EMBL EBI, which provide the data portal in ENA, and we've got all the partners and all their co-workers, and the Moore Foundation, Sarah Bender, John Kay, and Adam Jones. Thank you.